In this video, we're going to talk about copper redox or recycling. The definition of words in chemistry is a very important step in learning what you're being asked about. So you're going to consider the following terms and their general definitions. Be prepared to identify them in this experiment. This is an experiment where known amounts of reactants are reacted with each other to produce products, which are then isolated from each other by lab techniques. Which reactant is the limiting reactant and which reactant is in excess and will be determined mathematically. During the experiment, a rough idea of which reactant is a limiting reactant can be determined from the observations or what you're going to see happen during that experiment. These observations should be included in your digital lab notebook. So during the experiment, you're going to record both detailed observations and the number values to the precision of the measuring device that you're using. If you don't remember how to measure any device, make sure you ask your instructor before reading so you make sure you get the right amount of sig figs. You're going to read over the lab and report sheet before beginning the questions, and then the prompts about your experiment. And this can be done at home. You're only collecting data when you're in the actual lab. Now, we have a limiting reactant. The first thing you're going to do is you're going to write a balance equation for the reaction in the experiment. And then the next step is to convert the masses of each reactant into moles. Now, I know that we have talked about dimensional analysis. If you're struggling with dimensional analysis, make sure you check out the video library. There is a video on dimensional analysis. But really what I want to show you, is that we're going to take the atomic mass or the molar mass, which is your grams over moles, and you're going to need it to use it as a conversion factor. So if the copper 2 chloride used in the experiment was anhydrous or not, to know what to use. So anhydrous just means it's a substance and it contains no water. So you're going to convert from grams to moles, and you see how the grams crosses out, leaving the moles. That's your dimensional analysis. This approach works for a pure solid, but if your reactants were a solution, the volume and concentration would need to be determined the moles of the reactant. Slide is one of my slides that students love. This is straight out of the lab. You should be able to find it if you're looking in your lab. But this is just an analogy that I like to use and that the students seem to really enjoy. So this involves the making of a cheese sandwich. It takes 2 slices of bread and 1 slice of cheese to make 1 cheese sandwich. This is essentially a balanced chemical equation with the reactants on the left and the products on the right. 2 slices of bread plus 1 slice of cheese will yield 1 sandwich. If you have 10 slices of bread and 4 slices of cheese, how many cheese sandwiches can be produced? In other words, what is the product? So in this case, what is the limiting factor of making cheese sandwiches? The coefficient in the balanced chemical equation are in the units of moles and not slices. I know you guys know that, but I feel like I need to say it. So if you have 10 slices of bread and 4 slices of cheese, how many cheese sandwiches can be produced? The balanced chemical equation is the reaction relationship between reactants and products. So the number of moles of product predicted to be produced by each reactant can be determined. We're going to determine how much proper can theoretically be produced by the reactant, like the number of sandwiches. So we're assuming that the reaction between the reactants goes to completion and that our technique for recovering, isolating, drying, and weighing is perfect. Although this isn't always the case, this is how we're going to do this, okay? So the reactant that produces the least amount of product is your limiting reactant. All right, so you can determine the mass of product that can be produced since the moles of product from the limiting reactant is known. Your sample calculation, so if you can convert from grams to moles, you can convert from moles to grams. Note how the moles crosses out and leaves the grams, and vice versa. The grams will cancel out and leave the moles. Actually recovered by you and your partner is compared to the mass of the copper that the limiting reactant can theoretically produce. The percent yield can be determined. This is your percent yield formula. You can make sure you know where to find that because you may need it as we continue on. Examine these two masses to gauge how generally successful your experiment actually was. Make sure you are reaching out to your instructor with any questions that you have on the copper lab or copper redox or just redox in general, and how, also check out the dimensional analysis videos if you're struggling with conversions. And I hope this video helps you just a little bit to help you get going with your copper lab.